Okay, so this right here, we're going to have our quinones here. These can have, am I recording? Yes, I am. These can have 3 different oxidation states, 2 of which are actually stable oxidation states, and so it can last a fair amount of time. So this right here, this is the typical ubiquinone, okay? And that's the one that you typically are referring to when you say Q, ubiquinone, right? And this is the oxidized form here of that coenzyme Q, which is, again, ubiquinone. Is everybody okay if I say Q now? Okay, fantastic. All right, and so that one's the fully oxidized form right there. Now, if we get 1 electron stuck onto this being the reduced form, okay? So we accept an electron and a hydrogen. Again, the reduction of oxygen bonds to a carbon, that would be the reduced form. And so this will give us our semiquinone. Notice semi meaning partial, right? So semiquinone. This is actually an intermediate form. This is the one that is not particularly stable. This is not going to stick around for a long time, okay? By a long time, I mean like more than nanoseconds. All right. And so this is the QHI. Notice that little dot there. What does that mean? Typically that means free radical, right? Yeah. And so this is a radical actually. And this is one of the reasons that's so unstable. Okay? Now, again, QH dot, semiquinone. Now, the other way it can go is we can have an H plus come on here. Notice the free radicals on that other oxygen down here. You may not have noticed that before. That was down here. Okay. This is going to get another H plus added. It's going to lose an H plus, excuse me, off of it. And that's going to give you your semiquinone radical ion. Now, again, it's still a radical, but now it has a negative charge up here. Okay? And so this is going to be the semiquinone radical ion. This right here is going to be adding another electron and another H plus onto it. This is the further reduced form of this. Okay? This is typically what's referred to as the reduced form of quinone because the other form is not stable. It's radical. So that one's not sticking around for you to be able to go, hey, I have the reduced form of quinone. Okay? This, of course, requires you to have another electron and hydrogen to put on it, though, right? And so this is, again, that reduced form of the coenzyme Q. This is the QH2. This is the ubiquinol, okay? Notice the own versus here and all versus here. Why is that all versus on the right for the reduced form? Double alcohol, yeah. So the difference between them being you have the ketones, right? Own, alcohol, alls. Okay? Get both of the ketones converted to alcohols, get the reduced form. Anybody have any questions about that so far? Okay, so that cytochromes, they're going to be electron transferring proteins that contain a heme prosthetic group. They were specifically going to be focusing mostly on cytochrome C, though there are actually quite a few cytochromes, okay? You actually saw hints of that in the previous tables and stuff. So the cytochrome C, this is going to be a small soluble protein that's going to be loosely associated with the inner mitochondrial membrane. It's going to shuttle electrons from the complex 3 to the complex 4. It's present in all organisms with mitochondrial respiratory chains. It's going to be highly conserved across species because if anything happens to this, you lose the majority of your energy, right? Okay? And most mutations with this are not going to allow electron transfer. Electrons are very light, they're very easy to move around, right? So you can shunt them off into really different pathways very quickly, very easily. Cytochrome C from any eukaryotic species will react in vitro with cytochrome C oxidase from any other species. They're actually compatible in every species that's ever been tried. Okay? And why is that? Why would they be compatible? They're highly conserved. because if you do anything to them, it's very likely you are a dead organism, right? Yeah, you're not going to survive long enough to even develop, honestly. Okay. All right, so the three-dimensional structure of your cytochrome C is remarkably conserved among the distantly related species. This is tuna versus photosynthetic bacterium versus denitrifying bacterium. Very different species, diverged a very long time ago, right? You would not look at those and go, oh yeah, they're going to have the same protein there. At least I don't think you will. But now you will, I guess. Notice the structures of these. See how conserved that is? Note the important parts are these parts right here, right? The core. This stuff coming out is less important. Those are just useless curves that give it structure, right? Have to be there because you have to be connected. This part being the important part. See that? Okay, so the iron-sulfur clusters is going to play a critical role in reduction of reactions in biological systems. They're going to exist within iron-sulfur proteins. These are the non-heme iron proteins. Okay, notice these are different than the heme ones, which had that porphyrin crown, right? That you're sitting down on. Okay. The three kinds are going to be common in biological systems. You have a single iron atom. So you have a single iron ion. You have the 2Fe, 2S, and then you have the 4Fe, 4S. Notice what all those have in common. What's the ratio between iron and sulfur? 1 to 1. Yeah. That's why it's the iron-sulfur complexes, and then they're just, duplications. Okay. The iron ion cycles between the Fe2+, this can be a reduced form, and then you have the Fe3+, which is your oxidized state. They're going to undergo oxidation reduction reactions. They'll do that without releasing or binding protons. And so this is one of the nice things about this is that you don't actually have to move H pluses around, right? You don't have to get these H pluses moved around on this structure itself. You can transfer these oxidation reduction states without doing that. Unlike the NADH, FADH2, right? In which case you'd have to move the F pluses, or the H pluses, excuse me. Frataxin, oh shit, I forgot my water. A small mitochondrial protein that is crucial for the synthesis of the FES clusters. Friedreich's ataxia, this is actually a disease of varying severity that affects the central and peripheral nervous system, as well as the heart and skeletal system. It's caused by mutations in the frataxin. And so we have mutations here, and again, we're going to have difficulties laying out these, synthesizing these FES clusters, right? And so you're no longer laying your wire, and guess what happens to your system? It malfunctions, doesn't it? Okay, this is the single iron, and here we go on. Now, again, remember that the cluster itself, the single iron here, is going to have so when you have sulfur binding to the iron, okay, you're going to end up with a cluster of 4 in a sort of tetrahedral shape. Everybody see that? And typically what happens here is that when you add another iron, okay, you then have to form a bridge, and that's those 2. If you add 2 more irons, you have to again put 2 more sulfurs to form those bridges, okay? Now, it's kind of interesting that you could almost predict what's going to happen each step of this, couldn't you? So you have the iron here, it forms tetrahedral, and we can use the cysteines to form because we don't have to have anything in the middle, right? And so you don't actually have to add an additional sulfur there. This one, you have to have a bridge of sulfur between them, right? Or it won't be a single cluster. And so you have 2 sulfurs for your 2 irons here. This right here then is going to be 4 irons, And again, it has to be a semi-symmetric shape. It has to have something that's going to have, you couldn't have like a 3-iron here very easily. It'd be very difficult to synthesize that because, again, you wouldn't have a tetrahedral shape, would you? Yeah. And so you end up having an extra 2 irons, an extra 2 sulfurs. This actually makes this cage shape. See how it kind of makes a boxy shape? Okay. By the way, that's that same 4-membered ring. Notice it even has a little bit of a pucker to it that we have come to know and love. Okay? Okay. So the NADH Q oxidoreductase, this is complex 1, in case you forgot your complexes and how those relate. This is the proton pump that's going to serve as the entry point for the electrons from NADH. It's going to be encoded by genes in the mitochondria and nucleus. It's L-shaped with a horizontal arm in the membrane and a vertical arm that's projecting into the matrix. So remember that all of these are membrane bound in some way, and they'll project into the matrix in some fashion. Okay? They have to interact, most importantly though, with the membrane in some fashion. Okay? Complex 1 is going to catalyze this overall reaction right here. And so we have NADH plus Q. Everybody remembers that's the ubiquinone, right? And this is going to have 5 H plus ions in the matrix. The NADH is going to go to NAD plus. We'll get QH2. And 4 of those H pluses are going to go into the intermembrane space. So we've pumped 4. 2 of them jumped onto the Q, ubiquinone. And then we get the NADH going to NAD plus. And again, that's that NADH from our citric acid cycle, typically, doing this. So flavins are going to have 2 oxidation states. Your NADH binds, and the transfer of its 2 high potential electrons to the flavin mononucleotide, or FMN, prosthetic group of complex 1. This is going to yield the reduced form, which is FMNH2. The electrons are then going to be passed to a series of iron-sulfur protein clusters. They're in complex 1. And again, those are those iron-sulfur protein clusters we looked at right here. Okay? All right. And so right here, again, this is FMN here. And so it's going to gain 2 electrons, or we can say 2 hydrogens. Okay? It's essentially the equivalent thing here, reduced twice. And so this is going to then give you the flavin mononucleotide. This is the reduced form. This was the oxidized form here. Notice what has been added to are these nitrogens right here. Okay? Fantastic. By the way, this R group is still here. We just ran out of room. Okay? Or at least we ran out of patience. So membrane embedded part of your complex 1 is going to have 4 proton half channels. These are going to contain containing the vertical helices. One set is going to be exposed to the matrix and the other set to the intermembrane space. The vertical helices are going to be linked on the matrix side by long horizontal helix. Horizontal long helix is denoted as HL. Okay? You can kind of think of that as helix long or horizontal long. The intermembrane space half channels are going to be joined by a beta hairpin helix connecting elements. These are the beta H. And so the beta hairpin helix. Okay, so this is what this looks like. Last thing that we need to note is we have the Q chamber. That's this part right here, by the way. This is going to be an enclosed site where the Q accepts the electrons from your NADH. Okay, so again, notice how it's an L shape. Everybody sees the L? Fantastic. And so this part right here is going to be those beta H's right here. Okay. And this is going to be the part that's actually in the membrane space right here. Okay. This part right here has the Q channel. What direction does the Q channel come in from? Where's Q located? Was it located in the cytoplasm, the matrix, the intermembrane space? It's dissolved in the membrane, exactly. And so your channel actually opens up into the membrane, okay? And this is typically known as the Q pool. We'll get into that in a bit more detail later, but essentially it's just Qs that happen to be dissolved in membrane. Remember, these things are coming in and out all the time, okay? They're floating around in the membrane. Okay, and so this is going to go into the Q channel here. It's going to be an enclosed site here. This is actually quite important because the intermediates that we're going to make are charged, right? Many of those things are charged. And so we're going to have a lot of difficulties moving these things around if we try to do this in a membrane-like situation, right? That's very nonpolar. So your NADH is actually going to transfer that reduction or the electron density over onto FMN. This is actually going to transfer it through a series of iron sulfur clusters. You can think of those as your wiring, okay? And they work well because literally iron has very little orbital differences that electrons can jump past, right? Sulfur, when it's in contact with the iron like that, again, has really small orbitals that these electrons can come on and off fairly easily. Okay? And so we're going to have the electron density flow down onto our Q. This accepts two of those, right? So two hydrogens accompanied with electrons, okay? Or two reducing hydrogens. Now, this Q here, this is actually going to be used. The other thing we're going to have here is we're going to have this electron density, which is being pulled here. Remember that the overall reaction is going to involve the pull of that electron. We're going to have two of these hydrogens coming off onto Q, and the remaining 6 hydrogens, 1 on the NAD and 5 from the matrix, those are actually going to get pumped over, which is 4, is the remaining, is going to get pumped over into the intermediate space, intermembrane space, excuse me. Okay, and so this right here is these different channels right here. And so what we're seeing here is one of these actually comes through this part right here, which is right next to the Q. And this actually gets pumped out the other side over here. This one then is going to come into here, and is actually, we're going to have a release of the hydrogen H plus over here. And then we get a similar thing here, a similar thing here. Okay. And notice again, that is the, these are those subunits that we had mentioned up here. Okay. By the way, these being the hairpin loops right here, the beta H. Right down here. All right. Does everybody understand that this is the electron pathway right here? Okay. All right. So the Q is going to accept 2 of the electrons from the NADH. This will give you the Q 2 minus, 2 minus meaning reduced form of the Q, though it does not in the long run have a 2 minus charge, okay? At this point, it can have a 2 minus charge though, because we're inside the enclosed. So the Q2 minus is gonna have a negative charge. This will cause your conformational change in your LH and BH elements. And so this originally came in neutral. We got 2 minus charge. This is going to give you a conformational change in the rest of this. So this will lead into a change in the structures of your connected vertical helices that are going to change the pKa of the amino acids that are in that vertical helice. The H plus from the matrix is going to bind to the amino acids. This is going to dissociate then into the waterline channel, and it'll enter the intermembrane space. And so what we're getting here is we're getting conformational change the acidity. Remember, acidity is how well you take on and lose H pluses, right? And so we're going to take on H pluses here. This is then going to release this into this space over here. And then that's going to come out on this side. So the H plus from your matrix, the flow of the two electrons from your NADH to your coenzyme Q through the complex 1 will lead to the pumping of your 4 H pluses out of your matrix of your mitochondria. Pumping is kind of okay. Yeah, it attaches it to what is being used as a base, but it's not a particularly great base, right? And so this side then is going to have it release from that base. So Q2 minus is going to take up 2 protons from your matrix. It's going to be then reduced to the QH2. Again, that's still a reduced form. Okay? It's just not the negatively charged reduced form. This is what it looks like when it's the negatively charged reduced form, by the way. So this is going to contribute to the formation of your proton motive force. The QH2 is going to subsequently leave the enzyme for the Q pool. Remember, the Q pool is what again? Is it some special pool that exists in some fairy dimension? No, it's just your membrane, right? Yeah. So it can dissolve in the membrane now, right? Notice that it gaining this charge like this, the Q2 minus, it changes the enzyme, but what else happens here? In its negative form, could this go back into your membrane space and join the Q pool? No, it's charged, right? It's not going to go into a nonpolar environment. And so that actually does trap it here too, until we gain the hydrogens. Once it gains these hydrogens, it can release back into the Q pool or the membrane, right? And so it reverts back. So again, that's going to enter the Q pool. This is going to allow another reaction cycle to occur. The succinate Q reductase, this is complex 2. This is going to be an integral membrane protein complex. This is going to be in the inner mitochondrial membrane again. This is going to contain the succinate dehydrogenase. It does not pump protons. It results in less ATP being formed in the oxidation of FADH2 than the NADH would otherwise. Though the FADH2 has less potential energy in the first place, so they wouldn't have enough energy to drive the same sort of stuff the NADH does anyways. Okay? But yes, it not pumping that means less ATP in the long run. Electrons from the FADH2, this is going to be generated in the citric acid cycle. These are going to be transferred to your FES centers, and then you're going to transfer those to Q to form the Q2. Remember, in each of these cases, we're again transferring. Q is released and floating around from the later complexes, and we're going to transform those into QH2, which is again neutral, and so can go into our membrane. And we refer to those as the Q pool each time. Okay? And so the same Q is not necessarily used in any of these steps of the reactions. Okay? It could be any of the Qs, we don't care. Okay? We have a cycle of Q and QH2, Q being the oxidized, QH2 being the reduced forms. So the electrons from QH2, these are going to be passed to the cytochrome C, or site C. This is typically how you're going to see this from now on in every case. Okay? Is everybody comfortable with site C? Okay. This is going to be by the Q cytochrome C oxidoreductase. This is complex 3. So this leads to your net transport of 2 H pluses to the intermembrane space. Your QH2 is going to go with 2 site Cs. These are the oxidized forms in this case because they're going to take on the H's, the electron density. This is going to transport 2 H pluses from your matrix. This is going to give you Q that can rejoin the Q pool. And this will give you 2 site Cs in the reduced form. This will pump 4 H pluses into the intermembrane space. Hey, where did my extra H pluses come from? What the hell? Didn't I only move 2 from the matrix? Where'd the extra one come from? Got it. They came off the Q, right? And remember, you took in one of those from the NADH and one of those from the matrix, didn't you? Okay? And so now we're getting rid of them. Well, complex 1 at least. That was the complex 1 version. The complex 3 is going to contain 2 types of cytochromes. It's going to be named B and C1. It's going to have 4 prosthetic groups. These are 3 hemes. It will also have a 2Fe, 2S cluster. God, it's like I don't know how that happened. It did elite speak on me. Damn thing. Sometimes I really have to wonder about this. Anyways, heme prosthetic group. This is going to be in the cytochromes. This is actually an iron, a protoporphyrin 9. Okay, so here we have this wonderful, this side being matrix, this side being the other intermediate, intermembrane space, excuse me. This being the protein we've been talking about. And this right here being those iron sulfur complexes right here. This right here being the heme C1. That's located right there. And then this is the heme BL right there. And then this is the heme BH, and that's up here. And they're a little hard to see because they're kind of tucked in behind these rods here, which would be your alpha helices. Okay? But hopefully you can at least imagine they're right behind that. And notice we have our 2 major groupings of hemes, the heme C1 and the heme B, right? Okay. So the iron of the cytochrome, this is going to alternate between the Fe2 plus and Fe3 pluses. This is how you get the oxidized reduced forms each time, Fe3 plus being oxidized. The cytochromes will contain 3 hemes. You have 2 hemes, which are the BL forms. They're designated here being BL and BH to make them different, so you can understand one is here, one's down here. You also have a, you have the BL and BH here. You have the, okay, recollect. Me having no water is really messing with me. That was a bad idea. Okay, so again, we have our 3 hemes. I just totally skipped over the C1, didn't I? So the C1 is up here. This is the, we'll get to that a bit more later. But the BL and the BH are the ones that are going to be very different in these cases. And they don't seem different because they're the same B form, but their position dictates a difference. And so you're actually going to have the BL. The L stands for the low affinity. And this is actually 2 of those that are the BL forms. The BH on the other stands for high affinity. And this is the high affinity form right here. And these have to do with their structures and their positions here. Okay? So what's going to then happen is we're going to have the QH2. This is going to carry 2 electrons. The complex 3 electron acceptor, which is the cytochrome C, this only carries 1 electron. And so when you get a single QH2, this can't actually transfer both electrons at the same time to the same cytochrome C, can it? No. It just has 1 electron it can transfer at a time. And so your Q cycle is going to be this cycling of their mechanism. So the mechanism for the coupling electron transfer from your QH2 to the cytochrome C to the transmembrane proton transport. So in one cycle, 4 protons will be transferred out of the mitochondrial matrix, and 2 are going to be removed from the matrix. And so what we get here is we get a QH2 and a Q here. This is going to have 2 of these cytochrome Cs, which are oxidized. And again, remember, each of these can do 1. These right here can do 2 at a time. So this is going to have 2 H pluses from the matrix cycling up. Now what you'll notice here is that you get 2 Qs now. Notice that you've increased that number of Qs by 1. One of these Qs loses the hydrogen. This is going to leave a QH2. We weren't able to transfer both of these because, again, notice we only had 2 of the cytochrome C oxidized. This is going to give us 2 cytochrome C reduced, and it'll give us 4 H pluses that have been transferred to the intramembrane space. Again, remember that, yes, only 2 come from the matrix, but we can get the remainder from there, the QHs, right? This is where that preparation in complex 1 and 2 comes in handy, okay? This is part of where we're actually moving those hydrogens in the reduction, the H pluses, from our Qs that we generated before. Okay, so one reduced QH2, one oxidized Q molecule of Q is going to bind to the QO and the QI sites, respectively. So the QO, the QI. The bound QH2 is going to bind its 2 electrons through the complex. So one electron is going to flow to the 2Fe2S cluster, and then that will flow to the cytochrome C1, and finally to the oxidized cytochrome C, which will then reduce it. The other electron is going to pass through the 2 heme groups of the cytochrome B. This is going to be then to the bound Q. This will actually reduce it into that semiquinone radical anion. That's that highly reactive radical anion. That is a dangerous state, by the way, because it is highly reactive, right? So if you were to actually have it exposed to anything, this can react. Okay. This will, hopefully, luckily, please, stay tightly bound. Okay. If it were actually to drift off, it's going to bond to anything, right? Notice, by the way, this is that difference and why I kind of ignored this one before. Okay, so this is one pathway. This is the other pathway. Okay, I think I left off here. So the second QH2, this is going to bind to your QO site. It's going to have its electrons pass through the complex, as in the first half. One addition of the second electron on it. Did I just say one? Jesus. Here, I'm going to have to pause and get water.